Cultivation of Viruses, Part 1. In this video, you will be learning about the need to cultivate viruses and also the method for cultivating bacteriophages by using bacteria. Why should we cultivate viruses? Look at the situation in the world today. If we know something about the coronavirus today, it's because microbiologists have been able to study them. And these studies have only been done by collecting clinical specimens, such as maybe the body fluids of the corona patient, isolating the virus from those specimens, culturing them, identifying them, characterizing them. And with such cultures, it is possible to study the viral structure, the genetics of the organism. We can find out how they multiply within host cells and how the host cells are impacted by these viruses. Now, once we know the structure of the virus, we can also prepare vaccines. So, how do we cultivate viruses? We know that viruses do not have the machinery to make proteins or to make new DNA or RNA. They rely upon a host cell to provide them with all that. So basically, they hijack the host's machinery and make copies of themselves. For this reason, we cannot culture viruses as we do other organisms like bacteria. We cannot just plate them on a medium and wait for them to multiply. No, what we need to do is provide them with host cells so that viruses can enter into the host cells and replicate within them. And to do that, we need media for cultivating viruses. What are the media? The media would include whole organisms, embryonated, that is fertilized eggs, and cell cultures. So we can grow viruses within a whole living organism or within embryonated eggs or in an artificial environment that is outside a living organism, but of course within cells. This may be on an agar plate or a test tube or in a culture flask. Let's have a look at how viruses can be cultivated within bacteria. So those viruses that attack bacteria, they are known as bacteriophages, and most of our knowledge on viral replication comes from studies on bacteriophages. Let us say we want to make copies of this T4 bacteriophage that infects E. coli. So we have here a culture. We have here a broth culture of E. coli. We introduce this phage into this. Now the virus will infect the E. coli cells and replicate inside them. And this is an E. coli cell in which a virus, that is the T4 bacteriophage, let us say, has entered. It has replicated, and then the E. coli lyses and releases all these bacteriophages. Now, each of these phages is in turn going to infect other E. coli, another E. coli cell within this culture. And so the cycle continues till we have many, many E. coli cells that are lysed, E. coli organisms that are lysed within this culture. So because this is a continuous cycle, keeps on repeating, finally, as more and more of these bacterial cells lyse, the bacterial culture that was opaque because of the presence of bacteria initially, it starts to become clear. This is because of the bacterial cells lysing. Now that we know that the phages are actually there in large numbers in the flask, we want to harvest them. Now, one of the methods to separate out the virus from the rest of the culture is through this process called density gradient centrifugation. Now, the density gradient happens due to taking sucrose solution in different concentrations in a centrifuge tube. You can see that in this tube, there is a graded decrease in sucrose concentration from the bottom, where there is the highest concentration of sucrose, to the top, which has the least concentration. Now we add a volume of our sample to the top of this tube, and then we centrifuge it. Now when we spin this mixture, the different parts of it will separate. So for example, bacterial debris may be now found at the bottom part, and the virus may be found in a specific part of the test tube. Basically what happens when we do density gradient centrifugation is that the heavier components which are there, they will all settle down at the bottom, and the lighter components will be at the top. So once we finish with the centrifugation, the virus layer can then be pipetted out and used for further studies. Suppose we have an environmental sample, such as seawater. In that case, how do we isolate viruses from that sample? How can we find out how many bacteriophages are there in that sample? For example, in 1 ml of the seawater that we took, how can we find out how many viruses are there? So for this, we need to create a dilution series. So we can take about 0.1 ml of the sample and put it into 0.9 ml of some nutrient broth. Then you take 0.1 ml from here and put it into another test tube that contains 0.9 ml of the broth, and so on. Now this is somewhat like the pour plate method, where we had created a dilution series for bacteria. Now this nutrient broth in these test tubes also has E. coli introduced in it, in addition to the virus dilution samples introduced into it. Now we keep ready a nutrient agar plate with a solid medium. We keep that ready. And then from each of these test tubes onto separate Petri plates with nutrient agar on it, we just put this mixture which is there. So from this test tube onto one Petri plate with agar, this test tube onto another Petri plate with agar, like that. From each of these test tubes, we add the agar onto already present agar on those plates. So we already have solid agar on the Petri plate, and now we are adding some of this molten agar from each of these test tubes into different Petri plates. So that is what is known as top agar. So this broth that we have poured onto the Petri plate, it has got bacterial cells as well as diluted phage suspension. Then we incubate the Petri plates. And in the Petri plates, what is happening? Wherever the virus comes in contact with E. coli, it will penetrate E. coli, it will multiply within E. coli through the lytic cycle, producing more viral replicates. Then, as we have seen in the previous slide itself, the picture, the host cell will burst, releasing the virus particles, which will now infect more E. coli cells. Now, the places where the bacterial colonies have been lysed, we can see some clear areas, and these are called the plaques, the viral plaques. And the rest of this, so here these dots that you see, these are the clear areas and they are called the viral plaques. And this area which is in between these spotted plaque areas, that is the bacterial lawn. Now, wherever you see one dot like this, these dots, so these are the places where a phage has entered into a bacterium, replicated inside it, then the bacterium bursts, releases the replicates through lysis, then each replicate again infects another bacterium, causing its lysis too. So we can count the number of these plaques. And by counting the number of these plaques, we can work out how many viruses were there in the sample, in this sample, let us say in this initial sample. So we are basically assuming that one plaque represents one virus. Now, so you can see the plates here with different number of plaques. So these are the plaques here. These are the spots or the plaques. Now, in the serial dilution series, it's obvious that the test tube which had a higher concentration of viruses, it's going to be when we make a plate of it, when we make a top agar plate of it, this is how it's going to look like. And then when you keep diluting it, you can see that the number of viruses is decreasing because of the serial dilution, and that is getting reflected in these Petri plates as well. How can we grow viruses in plant and animal cells? We have just seen how we can grow bacteriophages within bacteria. So the next next topic relating to cultivation of viruses is how can we grow them within plant and animal cells, and also within cell cultures. So for this, we will be looking at succeeding videos.